AI can write essays, generate images, animate photos,
or even turn your voice into Taylor Swift. But all of this
is just surface-level stuff. Today, we're going to talk about the most groundbreaking, most impactful AI projects that will literally change the world. I'm talking like cures for cancer, digesting plastic, batteries that are many times more efficient, and more. So let's jump right in. The first project is AlphaFold by Google DeepMind. Now, the whole point of AlphaFold is to predict how protein molecules combine with one another and form these 3D structures, which has previously been very difficult to predict. And this helps with discovering and understanding proteins, which can help society. So, for example, this has huge benefits in biology, medicine, agriculture, and biotechnology. For example, protein discovery can help in developing drugs to treat diseases such as cancer, heart disease, and diabetes. It can also help with developing methods to diagnose things like dementia earlier. Or how about developing a protein that can digest plastic so we don't have so much plastic piling up in our oceans? There's currently so much plastic in our oceans that it's killing everything. Or how about developing genetically modified crops that offer higher crop yield, more nutrition, and more resilience so that we can have more abundance and reduce poverty in developing countries? All of this has to do with protein discovery and understanding how proteins work. The building blocks of all proteins are amino acids, and there are many different kinds of amino acids. Now, guessing how different combinations of these amino acids combine together to form a 3D structure has been very, very difficult. And this leads us to the Levinthal's paradox. Levinthal's paradox states that finding the native folded state of a protein by a random search among all possible configurations can take an enormously long time. That's because the number of possible conformations for a typical protein chain is astronomically large. If a protein were to sample all possible conformations sequentially, it would take an incredibly long time, far exceeding the lifespan of biological systems. However, that's not what we observe, right? We observe that once we throw these combinations of amino acids together, they merge into these 3D structures almost instantly. So the observation is that proteins do not explore all possible conformations sequentially. This would take an eternity. Instead, they navigate a complex energy landscape, rapidly transitioning between different conformations until they reach their native state. This is guided by the interactions between amino acids as well as the surrounding environment. However, previously, we were not able to figure out how these interactions or how the surrounding environment determined how these amino acids would merge into these 3D structures. For decades, researchers have used methods such as X-ray crystallography or cryo-electron microscopy to determine the structures of these proteins. However, these were very expensive and time-consuming. And for the past 60 years, using these efforts, we've only identified 170,000 proteins, while there are over 200 million known proteins across all life forms. So in the past 60 years, we've only been able to identify like a really small percentage of all the proteins that exist. Now enter AlphaFold by Google DeepMind. AlphaFold uses an AI model that was trained on tons and tons of data on how these amino acids would interact with one another in order to predict the 3D structures that would result from these interactions. They said on their first release, on July 22nd of 2021, they covered over 350,000 structures, including the human proteome. So this is already more than double of what we humans were able to accurately identify in the past 60 years, which was only 170,000. And this includes all the proteins found in the human. So once we understand how these proteins work inside us, there's huge potential in developing drugs, for example, that can help treat common diseases. So they go on to say that's all of the 20,000 known proteins expressed in the human body, along with the proteomes of 20 additional organisms important for biological research, such as yeast, the fruit fly, and the mouse. But this was just the beginning. In just the span of 1 year, on July 28th of 2022, they've expanded this database from nearly 1 million structures to over 200 million structures, including nearly all cataloged proteins known to science, which is just crazy growth. In the span of just 1 year, they covered almost all proteins known to science. So really quickly, I just wanted to graph this out so you can see how insane this growth is. Now, I found this chart, but it's not really accurate because it's not really to scale. So the final year in 2022, when they covered over 200 million proteins, this bar, you can see there's actually a break in the bar. Otherwise, if this were to scale, this bar would just shoot through the screen. So let me actually graph this accurately. So I'm going way back in time to roughly the same period, 1980. And then I'm increasing this by 5-year intervals. And then let's just assume throughout all this time, we've identified 170,000 proteins. This number really doesn't matter, as you'll see later. And then it says in 2021, I believe, so I'll change this to 2021, they've documented 350,000 proteins, which I'll have over here. And then in 2022, they've documented over 200 million. So I'll show you what this looks like to scale. You can see this final bar is just so massive that you can barely see the bars of the previous years. It pretty much looks like zero compared to this. And that's how crazy and impactful this project is. They say, by reducing the need for slow and expensive experiments, AlphaFold has potentially saved the research world up to 1 billion years of progress and trillions of dollars. So think on that for a second. If we were to use traditional methods to discover proteins, this task of documenting 200 million proteins would take us billions of years. But Google AlphaFold just did all of that in 1 year. So let's look at some real-world case studies on how AlphaFold actually changes the world. This paper uses AlphaFold to design an enzyme that could digest and recycle plastic. As you may or may not know, plastic is everywhere. We produce over 400 million tons of plastic every year. And the problem with plastic is it doesn't degrade. So it takes like over 300 to 400 years for a plastic bottle to fully degrade. And so we have a lot of these plastics, unfortunately, ending up in our ocean and killing everything from sea turtles, birds, fish, and even whales. However, we can't just end the use of plastic. There's a lot of things that depend on plastic, such as industries like food and medicine, because plastic is so durable and it's not degradable. So instead of attempting to just reduce dependence on plastic, this team at the University of Portsmouth is designing an enzyme that can break down plastic polymers so that they can be 100% recycled back to their initial state. This is one of the papers that they have published, and they used AlphaFold to help them design this protein. Another world-changing study that has resulted from AlphaFold is this one, where researchers used AlphaFold to understand the proteins that affect the health of honeybees, and then from that they were able to develop an effective malaria vaccine. Now, malaria has been a significant cause of death worldwide, particularly in Africa and other tropical regions. According to the World Health Organization, malaria caused an estimated 627,000 deaths in 2020. Now, since AlphaFold has a database containing predictions of all protein structures for all sequenced organisms, and this includes the complete proteome of the parasite that causes malaria, researchers have used AlphaFold to develop an effective malaria vaccine by understanding this malaria-blocking candidate called PFS 48/45. AlphaFold has also been used to accelerate the design and synthesis of a drug to treat liver cancer, specifically hepatocellular carcinoma, the most common type of liver cancer. According to the World Health Organization, liver cancer, including hepatocellular carcinoma, is the 6th most common cancer worldwide, and the 4th leading cause of cancer-related deaths worldwide. In 2020, it was estimated that there are over 830,000 new cases of liver cancer and over 790,000 deaths globally. And the most common type is hepatocellular carcinoma, which is a very serious condition that requires prompt diagnosis and proper treatment. So this was a study done by a team at In Silico Medicine Shanghai, which used AlphaFold to help them discover this new inhibitor, which can be used as a drug to treat liver cancer. Finally, AlphaFold has also helped with identifying hundreds of thousands of new psychedelic molecules. This could lead to the development of new kinds of antidepressants, which offers hope to those suffering from depression and other mental health disorders. So all in all, that is AlphaFold and just some of the many projects that it has helped accelerate. You can expect a lot more to come in the coming years. A lot more drug discoveries, a lot more enzyme discoveries that can help us break down a lot of different materials or help us with various industrial applications, and also new genetically modified crops that offer higher yield, which can help alleviate poverty. But AlphaFold is not the only project that Google DeepMind is working on. And this brings us to Google Genome. Similar to AlphaFold, which is used for protein discovery, Project Genome has been used to discover new materials. This AI was trained to predict the structures of materials with high accuracy. More specifically, they used this to discover millions of stable materials. Now, what on earth is a stable material? A stable material is one that maintains its physical and chemical properties over time under various conditions. For example, the stability of a material could be influenced by factors such as temperature, pressure, humidity, and other environmental conditions. But a material that is stable means that it doesn't change its physical or chemical properties when these environmental conditions change. So these stable materials are very desirable for various applications around the world because it ensures reliability, durability, and safety. So for example, you wouldn't want your phone suddenly blowing up because the material inside of it is unstable because of, maybe the temperature is too cold or the humidity is too high. You wouldn't want that to happen. So that's why we need to discover these stable materials, which can be used in our everyday products and applications. So a few examples of stable materials that you might be aware of include metals and alloys. For example, stainless steel is corrosion resistant and remains stable in various environments, making it very suitable for applications in construction, kitchenware, and industrial settings. Ceramic materials are also relatively stable and resistant to chemical and thermal changes, so they are used extensively in, for example, porcelain or certain types of glass. You can also find ceramics in dishes, insulators, and structural components. Plastic polymers are also stable in terms of chemical resistance and durability. So for example, these stable plastics are commonly used in packaging, containers, and various industrial applications. So hopefully that gives you a sense of what these stable materials actually are. So in this graph, it shows that about 20,000 of these stable materials have been identified up until now. Then we used some other computational methods, specifically from the Materials Project, Open Quantum Materials Database, and WBM Database. So with these improved computing methods, we've increased this number to 48,000. However, in the span of just 1 year, I assume, Google's Genome Project totally blew this out of proportion. So Genome discovered 2.2 million new crystals. That's equivalent to nearly 800 years worth of knowledge. How crazy is that? And then within these 2.2 million crystals, 380,000 are the most stable. So Genome discovered 380,000, and if you add everything together, I believe the total amount would be 421,000. Again, this is just crazy progress. It took humans years to get to this blue dot, and then just in the span of 1 year, Google Genome did this. So here they say this progress would be equivalent to about 800 years worth of knowledge and demonstrates an unprecedented scale and level of accuracy in predictions. So for example, 52,000 new layered compounds similar to graphene have been discovered. And this has the potential to revolutionize electronics with the development of superconductors. Previously, only 1,000 such materials have been identified. So with Google Genome, we've now increased this amount by 52-fold. We also found 528 potential lithium-ion conductors, 25 times more than a previous study, which could be used to improve the performance of rechargeable batteries. We'll definitely talk more about lithium and batteries in a second. Now, of course, you can't just discover these new materials and then do nothing, right? You need to look at ways of how we can apply these new materials in everyday applications. Like, we need to find a way to manufacture items with these new materials in order to put them to good use. And so they're already doing this. They have this autonomous lab in the Lawrence Berkeley National Laboratory where you can see there's this autonomous robotic arm, and it basically takes data from the materials database from Genome and uses machine learning to engineer new materials without any humans. So this is fully autonomous. You can see it's enclosed in this glass cage, I assume, just to make sure that if it triggers any explosions or any crazy reactions, it's all contained. Here it says, in a paper led by our collaborators at Berkeley Lab, researchers showed a robotic lab could rapidly make new materials with automated synthesis techniques. Using materials from the Materials Project and insights on stability from Genome, this autonomous lab created new recipes for crystal structures and successfully synthesized more than 41 new materials. And again, it's doing this all autonomously without any help from humans. These newly discovered materials can have potential applications in various industries. For example, they can be used to make better solar cells, better computer chips, and better batteries. Keep in mind, this Genome project was only published a few months ago in November, so it takes time for these new materials to be discovered and then deployed in the real world. But we should start to see these new, more efficient materials in everyday applications very soon. On the note of computer chips, a potential application of Genome could include inventing versatile layered materials which would be used in neuromorphic computing. Basically, these are like computer chips that are designed to work like the human brain. That's exactly what Rain AI is doing, and this is a company which Sam Altman is an investor in. They're trying to build a neuromorphic chip, or basically a computer chip that is designed like the human brain. And in theory, this brain chip would be a lot more efficient for AI and machine learning. So that's Google Genome. But Google's not the only one taking the spotlight here. How could we forget Microsoft? Microsoft published results on this project in late August, which I did a video on previously. And they did kind of the same thing. So they used an AI model to digitally screen over 32 million candidates, and they used an AI model to filter out 500,000 candidates which are deemed to be stable. And now the goal of this is they wanted to discover a new material for batteries. So they used further AI screening to filter these 500,000 candidates down to 800 candidates. Again, these are candidates that are deemed to be more stable and more suitable for batteries. And then furthermore, they used HPC screening, which means high performance computing screening. And this involves using powerful computational resources to analyze these large datasets or perform complex simulations to further filter down the candidates. So HPC screening is commonly used for material science and drug discovery. And so they've narrowed it down to, I believe, 18 candidates was the real number, all the way from over 32 million candidates. And they did this in less than a week. So here they say this case study highlights both the scale and speed of HPC plus AI solutions, as we were able to screen 30 million candidates in approximately 1 week. So here are some more numbers just to show you how crazy this innovation is. Traditional approaches require approximately 78 CPU hours or 4,680 CPU minutes per structural relaxation. In this internal study, our AI models required a little more than 3 CPU minutes, which is an over 1,500-fold speedup. So using AI, they were able to speed up this material discovery process by 1,500 times. So the numbers in this graph, I think they just rounded it to make it look nicer. They later specified that this is 32 million, and they've narrowed it down to 18 promising candidates that could be used in battery development in just 80 hours. And this brings us to lithium and the state of batteries today. Lithium is the main component in rechargeable batteries, and demand for this metal has skyrocketed in recent years due to their use in portable electronic devices such as smartphones, laptops, as well as electric vehicles. However, there's a problem with lithium, and we need to look for alternative solutions. Here's why. Like most things in life, lithium is a limited resource. And while current lithium reserves are substantial, there are concerns about long-term availability and sustainability. increasing demand for lithium-ion batteries could eventually lead to supply constraints and price volatility in the future. And the mining process to obtain lithium is very energy intensive, and it often causes lasting water and land pollution. Also, disposing these lithium-ion batteries raises concerns about environmental contamination and health risks from these toxic materials. So therefore, lithium is not a final solution, and there is a pressing need for a better alternative battery material. So these initial results from Microsoft, they were published in August of last year, which I did a video on, and then a few months later, in January of this year, this scientific article was published. Again, they talked about how Microsoft narrowed more than 32 million candidates for materials down to 18 promising candidates, and they found one that can reduce lithium usage in batteries by 70%. The new material is a blend of sodium, lithium, yttrium, and chloride ions, and it's basically a type of mixed metal chloride, and that was found to be the best option out of these 32 million candidates. And this statistic is insane. If this is true and this were to eventually be put into production, this would reduce our dependence on lithium by 70%. And discovery of battery materials is just one way we can use Microsoft's AI model, right? There are plenty of other materials that we can filter from this workflow. So this is just the beginning. Imagine how many new materials for how many different use cases we could potentially discover or invent from this AI project. It's definitely really exciting times. So we've talked quite a bit about material discovery. Let's switch gears to antibiotic resistance. This is among the greatest global threats to human health. Now, really quickly, let me just give you a bit of context here. Antibiotics are basically a class of medication that is used to treat bacterial infections. They work by either killing the bacteria or inhibiting their growth, thus helping our body's immune system fight off the infection. Common bacterial infections include UTI or urinary tract infection, or strep throat, which causes sore throat, and pneumonia, which basically inflames your lungs, and many types of skin infections. Now, yes, we can use these medications to kill off the bacteria. However, these bacteria can learn and evolve over time. And so antibiotic resistance is where the bacteria evolves and adapts to become resistant to the effects of these antibiotics, to these medications. Once it evolves to become resistant, then these drugs, these medications are pretty much useless or less effective. In other words, the bacteria are outsmarting the medicines that we create to destroy them. And like I said, this is one of the biggest global threats to human health. It was responsible for an estimated 1.27 million deaths in 2019 and contributed to nearly 5 million more. Now, this paper, just released 2 weeks ago, is really groundbreaking. It introduces SynthMol, which is a generative AI model that designs new compounds which are easy to synthesize from a chemical space of nearly 30 billion molecules. So you can see how this concept is very similar to Microsoft's project, which we just talked about, where they filtered down these millions of candidates to find a suitable material for batteries. Same thing with Google Genome. They've identified like 2.2 million materials and filtered it down to around 380,000, which were deemed to be the most stable. So here they have this AI model, which is able to design new compounds, and it was given this list of ingredients. It's a chemical space of nearly 30 billion molecules. And so they applied this AI model to design molecules that inhibit the growth of
Acinetobacter baumannii.
What she said. So A. baumannii looks like this under a microscope. It's a bacteria that can cause serious diseases in various organs. Some of the infections include blood infections, urinary tract infections, pneumonia, which is an infection of the lungs, wound infections, so if you get injured, the wound might get contaminated with this bacteria and it can cause a serious infection. Basically, it can infect a ton of areas in your body. So what this team did is they synthesized 58 generated molecules, and these were generated from this SynthMol AI model, and then they experimentally validated them, and from these 58 molecules, 6 of them demonstrated antibacterial activity against A. baumannii and several other bacterial pathogens. So there you go. Here's another example of the potential of generative AI to design new molecules to fight antibiotic-resistant bacteria. Here's another very similar article that was published in November of last year. So this is a team from MIT and Harvard, which used deep learning to screen millions of compounds for antibiotic activity. They then tested 283 of these compounds in mice and found that several of them were effective against the methicillin-resistant Staphylococcus aureus, and this other one, which I'm not even going to bother to pronounce. These two are the most hard-to-kill bacteria because they've evolved substantially to be antibiotic resistant. So the first one, Staphylococcus aureus, it's a type of bacteria commonly found on the skin and in your nasal passages. It usually doesn't cause any harm, but it could lead to infections if it enters the body through a wound. And then MRSA is a strain of this bacteria that is resistant to methicillin, which is a type of antibiotic. And because it is resistant to this antibiotic, it can be challenging to treat. There's only a limited number of antibiotics that are effective against this bacteria. And then moving on to this one, let's just call this VRE. This is a bacteria that is commonly found in our digestive tract. Now, vancomycin is a type of antibiotic that's often used to treat bacterial infections, including this one. However, there is a certain strain of this bacteria which has evolved to become resistant to vancomycin. And again, this causes it to be extremely challenging to treat this type of infection because there are only a limited number of treatments that are effective against this. But in this study, they were able to filter, like 12 million compounds and discover these new molecules that are effective against MRSA and VRE. Here's how revolutionary this AI is in drug discovery. If you think about traditional antibiotic discovery pipeline, it takes around 12 years to discover just one friggin' antibiotic. And then it takes between 3 and 6 years to discover any clinical candidates. Right now, with AI, you can discover in a matter of hours thousands or hundreds of thousands of preclinical candidates instead of having to wait 3 to 6 years. The team has been able to sort through millions of chemical compounds to identify a couple that seemed promising. So this is a common benefit of all the AI projects I shared with you today. The AI in Google Genome helps us save nearly 800 years worth of work. AlphaFold saved the research world up to 1 billion years of progress and trillions of dollars. Microsoft's project helps speed up material discovery by 1,500 times. So that basically sums up the top AI discoveries that are really world-changing. These all have massive consequences for the world, such as curing diseases, solving poverty, and giving us hyper-abundance and hyper-productivity. If there's one thing you should take away from this, it's don't die. Because things are going to get super exciting, and this is only the beginning. So, try to stick around for at least a few years. Let me know in the comments what you think of all of these projects, and if there's another project that I missed in this video that you think is noteworthy, please let me know in the comments below, and I would love to learn more about it. And if you found this video helpful, remember to like, share, subscribe, and stay tuned for more content. Also, we built a site where you can find AI tools and also look for jobs in AI, machine learning, data science, and more. Check it out at ai-search.io. Thanks for watching, and I'll see you in the next one.